All right. Good afternoon, everybody. Welcome to the latest seminar from the U.S. Society of Ecological Economics, continuing our series of putting the ecology back into ecological economics. Today's seminar is by Dr. Sam Purkis. His passion for underwater world was ignited at a young age by Jacques Cousteau, his iconic book and film, The Silent World. This early inspiration led him to become a certified scuba diver and eventually a diving instructor himself, living in a tent along the southern Egyptian Red Sea from 1992 to 1993. Coral reefs have since remained a cornerstone of his research endeavors. In 1994, Dr. Purkis pursued his academic aspirations in marine biology at the University of Southampton in England. His journey then took him to the Netherlands, where he earned both a Master of Science and PhD, graduating in 2004. Following his studies, he crossed the Atlantic to join the National Coral Reef Institute as a postdoc, where he rose to the rank of faculty. In 2016, Dr. Purkis joined the University of Miami as a professor, and a year later, he was appointed chair of the Department of Marine Geosciences. At the University of Miami, he continues to explore his diverse research interests developed over more than 2 decades while maintaining an active teaching schedule. Sam was the founding chair of the CCT-US, a position he held for 10 years, and he presently serves as chief scientist for the Living Oceans Foundation and for the recently launched conservation organization Odyssey, ODISEA. Dr. Purkis has authored 3 books and over 150 scholarly papers, several of which focus on ecology and geology of the Chagos Archipelago, challenging for my speech impediments, and his current research is centered on upscaling reef biodiversity assessments using new satellite technologies. So without further ado, I will introduce Dr. Purkis, and we're very thrilled to have you today. Thank you.
Brilliant. Well, John, thank you very much for the invitation. And I don't work a lot in the field of ecological economics. I'm really interested in what your organization does. And, it's a pleasure to speak to you. Since we're a small group, I mean, you can stop me at any time if you've got a question and there's something you want to discuss, or we can wait to the end. But please feel free to interrupt me. All right, then. So I'm coming to you from the University of Miami. That's the Rosenstiel School, which is situated on Virginia Key, and that's downtown Miami in the background. And please, I mean, if you're ever in the area, come and visit us and see the work that we do. I mean, we work beneath the waves, we work above them. This is the Marine and Atmospheric School to the University of Miami, which has a main campus, which is just sort of behind the city in the background there. So we're a satellite campus out there on the beach. To give you an outline of what I want to talk about, it starts with a grand tour of Earth's coral reefs. And this was a project I did with Living Oceans Foundation, their global reef expedition, which we finished some 10 years ago. But it was this huge data set that we collected, as I'm going to explain, that we've been working on for a long time. One of the areas that I really enjoy working is the Chagos. As you said, I founded the Chagos Conservation Trust, and that's a huge marine protected area which sprawls across about 1 million square kilometers in the central Indian Ocean. And you've probably heard of the Chagos because that's where Diego Garcia, the US air base, is situated. You have an air base out in the on one of the atolls there. So, but it's a much larger area than that. It also hosts the largest atoll on Earth, the Great Chagos Bank. Then I want to talk about how we start to think about biodiversity in the sort of work that I do on coral reefs, whereas we've been rather myopically focused on species in the last decades, and how we might upscale assessments of biodiversity using new satellite technologies. And there's a few really interesting things which are coming online now, and I'm going to have the opportunity to present a couple of our sort of new technologies which I see as the future leading us forward. And then I'm going to wrap up with some conclusions. So in terms of a grand tour, I was fortunate enough to be the chief scientist for the Living Oceans Foundation. This is a not-for-profit operating foundation, and we took their vessel, the Golden Shadow here, around the world on a 10-year expedition, visiting Earth's remotest reef sites and also mapping using the aircraft on the back. I mean, it's an incredible ship. It's got an amphibious plane that we can launch off the back of the ship, so we can fly hyperspectral surveys in really, really remote areas of the ocean. And this was a global baseline study. We started in 2006. So, as I say, this was not recent, more than 20 years ago. And we finished the field work 10 years ago, but really are still working up this incredibly rich and huge data set that we collected. I think it's fair to say this was the first sort of big data set collected for coral reefs. We visited 15 countries with the vessel, surveyed more than 1,000 reefs, and brought together more than 200 scientists on the different missions. And that project, we started in the Red Sea, and then we moved across for a couple of years to the Caribbean before going through the Panama Canal and the Galapagos Islands, and then a lengthy period in the 4 archipelagos of French Polynesia before leaving to the Cook Islands and Tonga, and then went through Fiji and New Caledonia and Solomon Islands, up through the Coral Triangle and the Great Barrier Reef, and then crossed and finished the expedition 10 years after we'd started in the central Indian Ocean, looking at the Maldives archipelagos and my favorite place, the Chagos, which is really just incredible because it's about as far from human impacts as you can get when it comes to coral reefs. And the mainstay of this work was standard sort of reef health assessments, the sort of things which divers are so adept at doing. And as I say, we had more than 200 divers involved through the expedition. So we collected a lot of sort of very basic data. For example, we did more than 9,000 transects counting fish around the world and benthic surveys, nearly 9,000 of those as well. And it's really easy to collect sort of high fidelity data in this way, albeit a little bit old fashioned. So we were looking at the biology, quantitative assessments of fishes and corals, pathogens, predators, algae and invertebrates, and of all the sort of key indicators on whether a reef is doing well and what its ecological trajectory is going into the future. We also looked at oceanography and chemistry, and being a geoscientist and a geologist for at least part of my career, we also were looking at the underlying geology using low fold seismic and so on and so forth. Now, this is great, but when you start working in this way, sort of diving on coral reefs and collecting very nuanced data, what strikes you is that the work you're doing is so drastically offset from the scale of the problems which are facing coral reefs. When we talk about changes in seawater alkalinity or thermal anomalies caused by heat waves, these affect reefs on regional, up to global scale, and very different from the tens of meters that you can look at when you are a scientific diver. And that's a problem, really, that we don't have a good way of looking at reefs with high fidelity at the scale that they manifest themselves across entire ocean basins. And this was a long time ago, 20 years ago, we started this expedition. And it was really sort of the dawn of a new technology, hyperspectral imaging technology. And we had an aircraft, which was like a great asset. And we were carrying it on the back of this incredible ship. And I proposed to sort of repurpose this aircraft and really fit it out with the state-of-the-art remote sensing toolkit that existed 20 years ago. And these were hyperspectral surveys and LiDAR and so on and so forth. LiDAR is a laser altimetry tool for working in the marine environment. So we took the aircraft and flew it back to the US, and we cut a lot of holes in the fuselage and mounted all of these new technologies. And it was really fortunate to be able to do that. The FAA got very excited when you start to cut holes in airplanes. They worry that you're going to sort of damage the structural integrity. So there was a very expensive and arduous recertification process. But we came out of it with really a very capable marine-based survey platform that we could take with us around the world. And some years before getting involved with this expedition, I'd done my PhD in the Netherlands, and I'd been really working on the physics of light in water. So we had the know-how to start to work with this new technology as it was then. And I've subsequently published a couple of books on that topic of how light in water behaves as it pertains to imaging the seafloor. And we started off with a sort of new way of looking at reefs at the time, where we could convolve new satellite technologies that were coming online with the hyperspectral airborne data. And this is an example from the Solomon Islands. That's Gizo Island in the top, and you can see the beautiful reefs in the turquoise color around that island. Then we could also map the bathymetry, so making a 3D topographic model of the seafloor that we could do optically from derivation from satellite, and then also using laser altimetry from the aircraft. And then we can push those two products together, the multi and hyperspectral imagery and the seafloor topography, excuse me, and we can start to map out what's going on the seafloor, just like you would do on land. And we created these habitat maps, as you see for this island, and the different colors represent the different coverages of the seafloor, whether it's sand or seagrass or coral, and what the state of the coral is. And we took that idea, these three different products, the high fidelity imagery, the bathymetry, and the maps, and we scaled it up to nearly 70,000 square kilometers. So we mapped about 1/4 of Earth's reefs, which was a really great start. Excuse me, bit of a cough. So, and this was an incredible opportunity because reefs are tricky in that they are obscured from casual view. I mean, even nations, I just came back from Indonesia, which is, has huge swaths of reef and some of the most biodiverse reefs on the planet. But at the sort of national, federal level, it's still not clear in Indonesia where all of those reefs are yet. And to conserve something, you've got to know where it is. And so these maps that we produced, starting about 20 years ago, these were, for many countries, the first accurate representation of where their coral reefs were located. And we could do this with high accuracy because we spent a lot of time ground truthing, so about 90%, and we could map to 36 ecologically meaningful habitat classes. And since then, there's been new ventures into this realm. So we did 1/4 of the world's reefs at 5-meter resolution and 36 classes. Later came the Allen Coral Atlas, funded by Paul Allen of Microsoft fame, which is coarser in resolution, 16 meters and only 6 classes, but that covers all of the world's reefs. So it's sort of built forward from what we were doing. And there's other initiatives as well, like this Millennium Mapping Project, which is, again, rather crude, 30-meter resolution and 5 classes. So we're now in a great situation, where you can go to almost any coral reef on Earth, and there's one or possibly several map products which exist to educate what's there and what the baseline status of that ecosystem is. It might seem rather archaic, but with our initiative, what we did is we took those maps and we compiled them into these large format, sort of coffee table type books. And I'm showing an example here from the Red Sea, another one from the Bahamas. And we went to each of those 15 host nations with those books. Why the books? Well, we discovered early on that actually having something physical and tangible that you can put on a minister or a prime minister's desk is what really engages them. Of course, all the data's online as well, but having this physical atlas that we could take started the conversation going. And that conversation began with every single conservation effort begins with a map. And we try to emphasize that this is the tool you need in order to understand what you've got to conserve. And of course, this was all at no cost to the host nations. And then we stood ready should those nations then come back to us and ask for advice about how to start to think about ocean conservation in the form of marine spatial planning or the declaration of marine protected areas. And some countries that worked really well in the Bahamas, in Tonga, in Solomon, in the Chagos, in the Central Indian Ocean. We got really, really large marine protected areas designated on the back of this work. And in other countries, we were less successful. We had good conversations, but we didn't, it didn't lead to real ocean conservation initiatives, or at least not yet. And we always stand ready to assist with that process. So anyway, that is that expedition. Of course, all the data are online as well for the community to use. And this, though, is not where we need to be. We're mapping habitat with living oceans in that case. We're not mapping biodiversity. So the top left there, you've got the habitat map. It's a great start. Shows you where the coral reefs are and the seagrass meadows and the mangroves. But it doesn't tell you what the divers are really measuring, which is the biodiversity in the ecosystem, the distribution of species, the number of species, and so on and so forth. And biodiversity is really, really important. And I know I'm preaching to the choir with your group, all experts on ecological economics, but the reason we care about biodiversity is it really matters, right? It matters for people, for planet, and for the future. And I start to think, for the work I'm doing on reefs, that biodiversity really consists of three pillars. We've got the habitat, the taxonomic, the habitat diversity, the sort of thing we're mapping from satellite. We've got the taxonomic diversity, the data which we were collecting with divers. And then most intriguingly of all, we've got the molecular or genetic diversity within the individual species. And I'm going to come back to that shortly because we're sort of working our way through those pillars. And biodiversity is nature's blueprint for resilience, and especially when we talk about reefs, the concept of resilience is so important because of the drastic shattering of this ecosystem that we've seen in the last days. So to frame the work that I've been doing, let me sort of give you this idea I have about the three pillars of biodiversity. And I literally think of it as three pillars holding up a bridge, going from the coarsest resolution on the left-hand side to the finest resolution on the right. And this is nothing particularly to do with coral reefs. I mean, I think this is sort of basic ecological ideas for any ecosystem. But with respect to reefs, on the coarse resolution side, in the first pillar, we have what we call habitat diversity. The arrangement of different habitats within a given area. So in my case, this would be seagrasses and corals and mangroves and how heterogeneous they arrange themselves. But when we work in the marine realm, just focusing on what's going on the seafloor is a rather blunt sort of approach to habitat. And now we start to think about it in a more holistic way, also considering what's going on in the water column above the seafloor. And we're coming to new technologies, and I'll present one of them where we can look at, for example, the temperature variance of the water over decades, but at much higher resolution than has previously been possible. And all of that will fold together into what I'm going to call habitat. Then the second pillar is actually the taxonomic diversity, the numbers of species and the distribution of species, our diver type data. And at the finer scale, the third pillar is the most intriguing and the hardest to access, particularly from satellite, which is the molecular or the genetic diversity within the individual organisms. Now, in terrestrial landscapes, in particular in forests and grasslands, there's been some really sort of foundational work which shows how these different pillars can be linked with ecological theory. So if you want to move from the habitat diversity to taxonomic diversity, there's the habitat heterogeneity hypothesis, pioneered in forests in the beginning, where you look at the heterogeneity of the distribution of habitats from satellite, for example, and the greater the heterogeneity, you presume the greater the creation of niches on the ground, and therefore you get a positive relationship between habitat heterogeneity and taxonomic diversity. And that's a well-established relationship on land, and I'm going to show you it works really well for coral reefs as well. Getting from taxonomic diversity to the third pillar, the genetic diversity, is a little bit harder because of the scaling that we'll talk about. But if you have great taxonomic diversity, you get amplified gene flow through the ecosystem, and that gives what we call ecotypes. Those are genetically distinct populations within a single species that have adapted to specific habitats. And it's been shown terrestrially, at least, that you can predict the probability of those genetic adaptations based on the quantity of taxonomic diversity you have in any given plot. And so there's a second link that we can then establish there through gene flow. If you can understand that adaptation mechanism, and that's what's so very important, you start to understand this adaptive capacity, that is, how can the ecosystem respond should conditions start to change. And if you understand adaptive capacity, it leads to the intriguing possibility that you might even be able to map resilience, which, at least in coral reefs, is sort of the holy grail at the moment, whether you could actually map adaptive capacity and resilience. So we're not quite there yet, but we're striving to get there. And I've been fortunate enough to have a couple of really good projects from NASA. One, the Marine Biodiversity Scaling Project, was part of NASA's biodiversity program, and Picogram is focused on individual coral organisms and how they grow and develop, was part of NASA's ecological forecasting program, that really funded us to try to look at these problems, like how can we link these pillars of biodiversity back to new emerging remote sensing technologies. And we were in a great position, I think, to tackle those questions because we had this incredible data set that we collected all the way around the world with the Living Oceans Foundation. So I lead that project, and Vay Chirayev, he's a colleague here in Miami, as well as Art Gleason. And really the work I'm presenting is Anna Baker. She was a PhD student in my team, and she's just graduated, and she did like a really fantastic job of unfolding a lot of what I'm talking about. So we had this incredible field data set that we'd collected in the old-fashioned way, going around the ship, around the world with a ship, putting in the hours, putting in the graft, and visiting all of Earth's remotest reef sites and surveying them with a reproducible, standardized methodology. And those are basic data, but they're really hard to collect, actually. And it was interesting because it set a baseline condition for the reefs that we visited in the years that we managed to get there. And that baseline is a great way now to track change going forward. And the sort of data we collected are as follows. I'm just giving a snapshot here from some aggregated island type data from across the South Pacific, but we have a measure of the live coral cover, which is a critical sort of health metric of any coral reef, the biodiversity of the corals, fish biomass, the biodiversity of the fish, fleshy macroalgae prevalence. Macroalgae is a problem on reefs, that's seaweed, in layperson's term, because it outcompetes the corals, and when reefs go into a phase shift situation, they become dominated by algae, and that is wicked because it stops the possibility of the ecosystem moving back to a coral-dominated state. So, these are the sort of standard, as I say, health metrics for coral reefs. And we started, with the NASA funding to try to make these links between the different pillars. And we were very pleased that it really works well. We took ideas initially developed in the terrestrial landscape and applied them to coral reef seascapes. And so starting here, this is one of Anna's papers just published a few weeks ago, where we looked at the habitat heterogeneity hypothesis from the very high resolution maps we've made from satellite and aircraft for a quarter of the world's reefs. And we showed nicely that we could retrieve the taxonomic diversity at those sites that the divers had been measuring. And we're not just doing that, in a boutique way at a few local sites. We're completing that derivation on a global transect around the world. So this seems to be a very robust relationship that we can use. A second paper here, this just came out, where we sort of even took a step back from that. So we used the spectral variation hypothesis, and we showed that you don't even have to make the habitat maps. You can actually use the spectral variability of the remote sensing data, and that also proxies niche generation, and you can reproduce the taxonomic diversity as counted in the field by divers. And a third paper of her thesis, which was published a couple of years earlier, we take a step even further back, and rather than just looking at the seafloor now, we are looking at the holistic definition of habitat, so the thermal regime of the water, human influences, hydrodynamic flow, and the seafloor heterogeneity. And again, we show that you can reproduce a taxonomic diversity on the seabed with really well. Now, I don't want to go into too much detail on how we do that proxy technique, but there's several ways that you can use remote sensing data in this way. And we found that ensemble learning methods like random forests were very successful, where you can feed in multiple predictor variables to get your response variable. In this case, it could be the biodiversity of corals or fish. And we also showed even regression-based models work fairly well in this regime as well. So we've got many different workflows to accomplish the task that we're trying to do. So it would seem that already with existing remote sensing technologies, we can proxy taxonomic diversity at global scale based on what we can map from satellite. So that's great. And I'm really happy with the outcome, but it's not where I want to be. I mean, I really want to get to the stage where you can actually predict genetic diversity of the individual within the individual species as a stepping stone to mapping adaptive capacity. And I think for reefs, at least, if you think about if there's one physical parameter which is well understood to moderate genetic diversity, it's temperature. Corals are strange in that they're particularly susceptible to rising temperatures. They live very close to the upper thermal tolerance, which is a pity for them, because even small changes in ocean temperature can lead to mass bleaching events and mass mortality. And that's how we've lost so much reef in the last decades. And there's an ample chain of lab experiments and field experiments that show that corals, if you expose them to high temperature, but not enough to kill them, but enough to stress them repeatedly in a sustainable way, those corals possess the alleles that code for proteins which are better able to tolerate heat shock, better at least than their counterparts which are in cooler waters. And so if you can map what I would term thermal microclimates, you can hopefully predict where you get these genetic adaptations which are going to be well poised to deal with future ocean warming scenarios. And the way to map thermal microclimates is the first sort of new technology that I want to present to you. And so it's all to do with temperature. And like, first of all, what is the state of the art for understanding temperature in the oceans? Well, NASA has the GIRST, Group for High Resolution Sea Surface Temperature product. It's released every 24 hours and has a resolution of 5 kilometers, or 1 kilometer in its experimental version. And it shows the temperature distribution in the oceans on any given day. And it's fantastic. I mean, I'm just showing a snapshot here from a few weeks ago. And if you want to know what the temperature is in the South Pacific, you can see that, perfectly at regional scale. And it is useful, these sort of products, for predicting whether a coral reef is going to bleach, at the scale of the entire Pacific Ocean. But these sort of state-of-the-art data have a real problem that 5 kilometers is a very crude spatial resolution. And we already understand when it comes to coral reefs and watching them die, unfortunately, that even on scales of hundreds of meters within an individual coral reef, you get huge variation in bleaching response and mortality response. So there's very fine scale nuance to this disaster which is unfolding. And I can emphasize the problem with these existing data. I'm going to take you to another one of my favorite places to hang out, which is the Red Sea. I was just there in January, actually, doing some work. And well, I've been working there since I was a diving instructor in the early 90s. So I know the Red Sea is a very special place to me. I love it. And there's a great university north of Jeddah on the Saudi Arabian coastline, the King Abdullah University of Science and Technology, KAUST. You know, it's sort of like the Ivy League of Saudi Arabia. And there's a great collaborator I have there, Francesca Benzoni. And offshore the university, there's a whole series of reefs stepping offshore, which are really well instrumentalized. It's a perfect sort of natural laboratory to work in. And I'm showing in the middle and the bottom pane there this standard 5 kilometer. No, this is a 1 kilometer, even the highest resolution experimental product, temperature product that you can get globally. And you see, you don't have a lot of fidelity. And worst of all, when you get close to the coastline, there's no temperature retrievals from the satellites at all. And that's where most of the reefs are located. So we don't even know their thermal regimes. And the reason for that is the emissivity of the water versus the land is so different. We can't close the algorithm to reproduce the sea surface temperature. So the coastal areas go unmapped. So this is not good. This is a disaster when you really want to understand what's going on at the scale of individual reefs. So not being deterred, we got creative. And we started now to use the International Space Station as a satellite sensing platform. Of course, the space station's famous for lofting astronauts and cosmonauts, but also on the upper right arm of the space station there, on the upper limb, there's a module where various experimental radiometers are attached. And one of them that we became particularly excited about is this module called EcoStress. And it was designed to look at high resolution temperature, but on land, particularly as it pertains to rainfall around the world. And we had the idea that that might be repurposed, that radiometer, to actually work in the water. And it does. It works really well. And this is a PhD student of mine, Jake. He's starting this project. And he's shown, and this is those same reefs offshore Saudi Arabia, the middle is the state-of-the-art NASA 1 kilometer product, and the bottom pane is our new product, which is 70-meter resolution. And so that's 250 times finer fidelity than we get with the existing NASA data set. And you can see in the bottom right there, we can now understand the variance in temperature on scales of just hundreds of meters. That is what's going on within an individual coral reef. And I'll give another example of that. This is what we call a map of thermal microclimates, where the different colors here denote different thermal regimes over a number of years. This is the Al Waj Lagoon. It's the largest lagoon in the Red Sea. And there's two things to note. First of all, that even around individual islands, and you can see the blowouts on the right-hand side there, there's very different thermal settings that the corals are experiencing. And the second thing, because we have this ultra fine resolution now, we can also map right up to the coastline. And so we can audit the thermal regime of corals, which are living close to islands or the coastline, which go completely unmapped using the old technology, what I'm showing the equivalent here with its very, very crude fidelity. So I think these new technologies are really valuable in that we can start to predict coral bleaching, like I'm showing here from the central Indian Ocean in the Chagos archipelago. It's been ravaged by bleaching. But we can start to predict bleaching at scales of hundreds of meters, not scales of entire ocean basins, and then dedicate scarce resources to the reefs, which are situated in the regimes which are most dangerous for their continued survival. And when we start to designate new marine protected areas, we can also select a portfolio of reefs to protect, which have hopefully sort of built-in adaptive capacity because they live in thermal regimes which give the necessary genetic adaptations to heat shock. And
Sam,
that work,
Sam,
oh, yes, yes.
Could you, Karin Lindbergh here, could you please go back to the previous slide for a second?
Wait, I go the wrong way. There you go.
Yeah, yes, that one there. Is this saying that the eco stress underestimates the temperature?
Eco stress systematically underestimates the temperature by about a degree.
Yeah.
But this is a well-known bias, and you could just correct for it because it's
Yeah.
systematic. It's
Okay.
it's due to how the temperature is derived. So it does slightly underestimate it, but we could correct it easily.
Yeah. Okay. Thanks.
Yeah, yeah, yeah. Okay, so that work is that works. Jake, my PhD student, he just published that work. Well, yeah, just 2 weeks ago. So congratulations to him. All right, so I think with that technology, and I've got one more to present to you, that we are getting to the stage now, at least when it comes to temperature, that we can look for genetic heat adaptation. And certainly we know where now to sample, and that's what we're doing. So we're sampling and sequencing corals in different thermal microclimates. We've got a project to study in the Philippines, and we're working in the US as well, to see if we can predict the prevalence of those specific adaptations. Now, the final technology I want to present to you is working more on the taxonomic diversity, the sort of information that divers can collect. But rather than just working at tens of square meters like a diver can do, I'm trying to rapidly scale up now to hundreds of square kilometers. And this technology is called the Fluid Cam. It was developed by a colleague of mine, Vaitrayath, here in Miami. And it's a drone-based instrument at the moment, but we hope to test it in orbit in the next 12 to 18 months on a small sat sort of experimental launch. And the Fluid Cam operates by using what's typically considered to be a noise as a signal. And that is the ocean waves. And you can see here in this video, it's just a test tank here, that as the wave moves across the sea surface, it acts like a lens, hence the name fluid lensing, and it magnifies and demagnifies whatever target is beneath the water. In this case, it's a series of corals. And if you catch the magnification events, you can image at very, very high resolution as compared to if you don't do that, where inherently any image taken through water, which doesn't account for this lensing effect, is, physically blurred. But here, we're actually using the lensing to sharpen the image and get incredible resolution. And we really have to understand that lensing. And this is a computer simulation in a NASA test tank, nearly 50 million CPU hours here. And we're looking at that lensing effect, and I'm slowing it down here. And you can see those magnification, demagnification processes. And we have to build up an understanding of how that operates at different water depths. And once you understand that, the results are really actually quite staggering. So this is data from Vade's team collected out in Guam, and that's the raw drone imagery. And then you apply the lensing effect, and it's effectively sort of mathematically removing the water. But because we're lensing, the pixel resolution now goes down to about millimeters. So it's incredibly powerful. We also get the topography as a byproduct of the lensing. So we don't need LiDAR and so on and so forth. And then we can start to map that imagery in an automated way using neural networks, which is an exciting avenue. But the problem with a neural network is it uses a lot of training data, and providing the necessary training data is a problem. So we have a gaming app available on the Apple App Store, where citizen scientists are then tasked with interpreting this imagery, and we sort of gamify it. So the better they get at it, the more elevated in the game they become. And we've got tens of thousands of people now sort of working in this gaming environment, providing really excellent training data that we can now use to quantify and classify that fluid lensing data. And all of that training and analysis is carried out in NASA Ames, because we need a lot of compute at the moment to do that. So from my perspective as a reef remote sensor, this is really a step change in how we look at reefs from above. On the left-hand side is of the current satellite state of the art, which can be sharpened in the middle pane there, at least in a panchromatic sense, but you don't see much when it comes to individual reefs and individual corals. And then on the right-hand side is the fluid lens data, which brings us down to millimeter fidelity. You can actually start to identify individual corals to the level of genus and even species. And through repeat surveys, you can track the growth and death of individual coral colonies. So this is scaling up. Maybe divers could look at such parameters through time on a handful or several tens of colonies. Using the fluid cam, we can now scale that up to tens or hundreds of thousands of colonies to really get the parameters that you need to drive ecosystem models for reefs. And we can also use these data with a different processing pipeline to count the fish. So we can get fish biomass and fish species diversity also from the fluid cam, again, alleviating the expensive and logistical constraints of deploying divers all around the world. All right, then. So I think this is a lovely example of how academic and philanthropic and federal partnerships can work together. So we had some ideas at University of Miami, and then we come to the Living Oceans Foundation, and they lend us their vessel kindly, and we go around the world for 10 years and collect the data that we need, the basic data. I also set up a charity, the Chagos Conservation Trust in the US, which allowed us access to some really remote sort of untouched reef sites in the Indian Ocean. Then we go to NASA, we have a Space Act agreement with them, and we get the necessary funding to really build out the new technology. And hopefully that's now putting us on a path to testing that new technology, not on drone, but in orbit in the next 2 years, all being well. So in conclusion, I've shown you that reef habitat can be mapped at scale, global scale from orbit. We started that sort of 20 years ago. Still, now, as it was then, every conservation effort begins with a map. This is just so important in the marine realm. And for areas like the Chagos, which is my happy place, the need for mapping has never been more pressing, to set a baseline, because degradation is happening so quickly, even in the remotest parts of the ocean. We need a baseline to set what our conservation aspirations should be. And you've got other derivative products like blue carbon and so, which also benefit from rigorous mapping. Biodiversity is really important for resilience, and I've shown that I think about it in terms of habitat and taxonomic and molecular or genetic diversity, and they build together into these three pillars. And I really feel confident for the marine realm. We're very close to having remote sensing solutions now to all three of those pillars. And it's about time, because as you know, as well as I, biodiversity really matters for people, for planet, and for the future. And I'll just say, as my final slide here, that I'm a coral reef scientist, and it's quite a depressing work. We've lost about 50% of the world's coral reefs in the last 40 years, which is a terrible statistic. And we're probably going to lose a meaningful proportion of what's left in the next 10 to 15 years. This is what we call the reef crisis. It's important, though, to remain positive. There are signs of adaptation, albeit perhaps not quick enough. But I think it's really important that these new sort of technologies we're developing are deployed at scale and not left languishing in the lab, because they can be very informative when it comes to understanding the distribution of resilience. So that's really the push of my team at the moment, is scaling those technologies and getting them deployed as quickly as possible. All right, then. Well, that's that. I'm happy to answer any questions if you've got any. And it was a great pleasure. Thank you so much for inviting me.
No, thank you, Sam. That was excellent. Great presentation on the technologies and the issues with coral reefs. There is a question. John Sorrentino, you want to go ahead and ask yours?
Yeah. Have you considered linking adaptive capacity with prescriptions for genetic species modification for reef preservation? I think I read something about people are doing genetic modification to reduce heat stress in certain species, etc.
Yeah. We have projects in Miami doing that. In our marine biology, we do a lot of work on reefs. Yeah, the trouble is the scaling. The trouble is the scaling, and I think it's been shown reasonably convincingly that you can breed corals and manipulate them to make them more resistant to stress, such as temperature being the prime one. And you can even actually sort of modify them using inoculations for pathogens or so once they are in the adult stage. The problem is that the corals, they just grow so slowly, and even sort of outplanting relatively small areas like hectares. There's a project in Saudi Arabia. I mean, that's where the dollars are after all, where they're outplanting some tens of hectares with new sort of modified corals growing like you describe. And the project has a budget of $1 billion. $1 billion, I said. And, if successful, that will deliver a sort of modest reef in a very large ocean basin. So it's a great technology, and it's one that should be pursued, but it's a scaling problem. And the fact that the corals, they just grow so slowly, and they're very susceptible at certain life stages to changes in their environment, and sort of getting success of outplanting lab-grown corals, the mortality rate after 2 years is very, very dramatic.
Oh, thank you. I have one more question, if I might. I have a student doing a research paper on degradation of coral reefs. May I give her your email address?
Of course you can. Yes.
SXP. Good. Thank you.
Yeah, that's it. I've got a more common email address. I mean, if you just Google me, you'll get it. But Miami has this rather ugly email system. Yeah.
Oh.
All right. Next is Tanya.
Hi, Sam. Thank you for the amazing presentation. It was really, really interesting and well delivered. I have 2 questions, and maybe I missed one of them. The first one was with regards to the accessibility of this data. Is there a vision to have a central repository where this data can live and be accessible? Are there any ideas of the cost? Is it meant to be a publicly accessible data, or maybe a membership-based or a purchase-based model? So that's my first question.
Yeah, no, absolutely. I mean, no, there's no. It'll be public domain data. It's here to be used. I mean, for these new high-resolution thermal products that we're developing, they're not community ready yet. We're starting to meld in some other sensors beyond the space station to get better temporal resolution. So we get a look every 4 days, we hope, when we finish that. And then the hope is that this will become a standard NASA product, and so it would be, communicated through federal channels, and/or we have our own sort of data portal that we can also push those products out with. So we very much, want to share it, but at the moment, for the thermal data at least, we still want to make sure that what we're sharing is accurate and reliable.
Mm-hmm. And the second question, I actually work for conservation finance, and we are constantly making the business case to investors and why invest in nature. And we always have a hard time translating very academic, complex measures and metrics and indicators into kind of meaningful for them, let's say, impact indicators. I was really glad to see what you said was the standard health metrics, and I think you presented like 5, and I assume those are the metrics we can go to and track across projects. Are there any other initiatives that tend to maybe simplify even more this information as a communication, kind of thinking about packaging the information for the investor community?
Yeah, I mean, that's a super question. And that group that I'm involved with, ODISEA, ODISEA, that initiative is very much what we're trying to do. So this is a global survey of marine biodiversity that we're trying to accomplish in the next 6 years. I mean, it's sort of like doing the Global Reef Expedition again 20 years later, but with more modern technology. I mean, that's really my vision. And the idea here is that we could do a lot of those standard sort of ecosystem metrics. We can now move that to orbital observations. So you could take any area around the world and you could almost have a dashboard which is tracking at very high fidelity, changes in the seafloor configuration, changes in the thermal environment, and even predicting taxonomic or genetic diversity within those areas. And I think, this is probably more your expertise than mine, but when it comes to ideas like biodiversity bonds and so on and so forth, having reproducible orbital-based eye in the sky type dashboards of keeping track of large swaths of the coastal zone. I mean, you could say the same for terrestrial landscape or the open ocean, but I'm interested in the coastal zone. That could be really, really interesting. So the idea is to sort of take the need off of exhausting field work and do a lot more from orbit.
Right. Next is Corinne.
Hi, Sam, that's amazing stuff you're doing. That's quite remarkable. I want to follow up on Tanya's questions because I'm leading a seminar this semester on basically evaluating ecosystem services generated by forested properties at my small environmental college. And when it comes to biodiversity, I've been, just really wondering how does that translate? How is that something that you give people a sense of? I mean, as an ecologist, of course, I know it, but how in a world where you're trying to kind of put it on parity with a development, let's say, how do you square that? How do you convince people about that? And it sounds like Tanya's kind of on it with some kind of biodiversity funds. I'm going to listen to a webinar next week sponsored by the Ecological Society of America on biodiversity. What is it? Investment funds or something like that? I forget. I'd like to just hear anybody's thoughts on it, Sam, anybody else. I have to hop off pretty soon, but I'd love to hear a few people make a few points.
Well, I'll take a quick grab at it. I mean, it's really difficult, Karen, and we experience exactly that you do. That trying to convince that biodiversity is foundational to sort of like everything that we are and what we stand for is a challenge. And it's one of those things, wasn't it, Joni Mitchell, who said, you don't know what you've got till it's gone. It's very hard. I mean, I think from my perspective, I'm also a geologist, and we see in sort of Earth's history play out time and time again the complex systems and reefs are a classic complex system that when they are perturbed, they take a long time to regenerate and rebuild. And by long, I'm talking millions or tens of millions of years in many cases. But complex ecosystems are so incredibly valuable because of the biodiversity they host and all of the ecosystem services they provide. But I mean, just taking, and this is probably a sort of off the wall example, look, from a geologist perspective, these complex systems are the products of incredibly long series of uninterrupted evolution. And if you break them, it takes so long to get them back again. I don't know how to convince someone that biodiversity is as important as development. I mean, that is the $1 million question. I'm happy for anyone else to sort of grab it because, we just try and be as convincing as possible, but it doesn't always work.
Exactly. But I mean, I think that, we've got to get away from just money as the sole thing that people pay attention to, and that's I think that's our quintessential struggle.
Yeah, I mean, I completely agree with you. And for reefs, it's a struggle against a timeline, because if we keep arguing and struggling for too long, there isn't going to be any to worry about quite soon.
Definitely. I work on ocean deoxygenation. I hear you.
Yeah, yeah,
Now we have Mr. Trump to deal with.
we certainly do. Yeah,
Aren't we lucky?
yeah.
Tanya, did you want to go ahead and speak on what you wrote in the chat?
I mean, that was pretty much it. I hear you, Karen, as we often have to make this point in our outreach to investors and in making the business case. And at least the realm I work in is translating things like the health metrics into ecosystem service value. So the non-market benefits of these conservation goals. So just
And
make
how do you tell when you've actually convinced somebody?
we get money.
Aha. So there is a way. So that is very interesting. What I would love is for I'm at the State University of New York, and what I would love to see is the State University of New York actually appreciating the resource treasures that we have, which they don't at the moment. They see it as a liability, and I think they should be seeing it as on the other side of the ledger. So that's kind of my take on this. But this has been fascinating. I think we ought to have more talks about these topics. But Sam, I really want to thank you for a wonderful webinar. And I'll have to hop off now.
Me too. I've got to hop as well, I'm afraid. Look, it was really a pleasure. Please, John, circulate my email around, and I'd love to continue the conversation, and come and visit Miami. We'd love to host you.
We will certainly take you up on that, and make sure if anybody has any questions to share your email. I know I have one, so I'll be sending you something. And thank you so much for a very interesting and fascinating seminar.
Great. And, particularly if you could continue the conversation about financial, instruments, because it's something I'm becoming interested in, but I just don't have any expertise. But I'm really, really interested in how this sort of work can play
Check
into
out
that
this
sort of work.
ESA. So, if your email comes, I'll send you the link to the ESA webinar next week, because it's free for anybody.
Karen, I'd really appreciate that. I really appreciate it.
Well, with that, I just quickly will share next week's seminar is Ecosystem Accounting of Coral Reefs of Hawaii. And our speaker will be Kirsten Olesen. She's following up on the science component that we got this week with the economics component. So we are trying to do that linkage as much as we can, and because it is important. So with that, thank you, everybody.
All right. Thanks a lot, Sam.
See you soon. Thank you. Bye.